interesting guest for this week. It's Dr. Robert Gallo. He is the Homer and Martha Gudelsky Distinguished Professor in Medicine. He's the co-founder and director of the Institute of Human Virology at the University of Maryland School of Medicine, and he's the co-founder and international scientific advisor of the Global Virus Network. And the reason we want to talk to him, this is a very famous scientist who is the co-discoverer of HIV. He's the guy who discovered the HIV blood test. He discovered HTLV, which is one of the first retroviruses that causes leukemia. And so he's playing an important role in the research of COVID. And we wanted to ask him a bunch of questions about what he's up to and what we can expect from some of these other vaccine ideas that are coming out. And that's coming up right now.
Yes, I've been in science for, about 55 years.
Wow.
I did my first experiments, when I was 19 in my garage in my home.
Wow.
With some mice back. I decided, I read a pamphlet. This is one thing I had was always dare. They said no one knows the function of the thymus gland. I said, this summer I'll find out. And so I, had no research experience whatsoever. My first year in college, I sent all my surgical equipment home, sent my mother around 200 mice, and
oh my God.
Kept them in the garage for about 6 weeks till I came home. And I learned three things. I dissected to get the thymus out, right? And see what happens to the mouse. And I found out they all died, 67 in a row. So I said, boy, the thymus is really important. But of course, I cut the aorta, arch of the aorta in all of them, right, and over the heart. And I learned three things. I would never be a surgeon. Number two, you really need to walk before you can fly. You need some technology. Even the journalist must learn to be able to scribble, right?
That is true.
You must be able to think when the BS is coming and when it's real, etc. And the third thing is I learned my little Italian mother was not so always so sweet and kind, because one day the mice all disappeared.
Really?
I don't want to say what happened to those mice. Yeah, she got fed up because the stink was everywhere, and I was getting nowhere and throwing things around actually, and not very happy. And so the mice just simply went away. And it took years before she told me what happened, but I don't want to tell you what she did. It's really
Did
bad.
she cut their thymuses out?
She did. I don't want to tell you.
Was it painless?
She just had this little smirk and grin on her face. Okay. What she did was she obviously got rid of them, right?
Right.
And, and it was only 18 when I did an autopsy. I mean, I wasn't even interested in science or medicine or anything else. I was interested in basketball and girls, really, to be honest, but then we don't have to write that down. Then I had a bad experience, very, very bad experience. My sister had a very leukemia and my parents left, and I didn't see my parents for almost 2 years. And then she died. She died miserable, and I saw her before she died. It was a horror show. And then my father, I was, just into basketball and so on. And then my father really kind of cracked up for years, and he became friends with a pathologist who made her diagnosis. And that pathologist took me under his wing. He was the first rationalist cynic I ever met. He was critical of doctors and very rational, quizzing everything. I never saw that before in my family. And I got close to him. And he rarely smiled. He didn't have any sense of humor at all. But one day there was an unclaimed body. He said, "You've seen if you do it." And he walked out. So I tell you, that was a horror. That was really awful. I mean, you forget the mice. If you're with a dead body and you got to do the complete dissection and explain why he died.
So did you figure it out? It was up to you to.
Yes, at 19 years old. Yeah, what was it, 18 or 19? I don't know. It was the same age. It's probably the same summer. It was 19. Yeah, it was not easy.
I mean, I was reading about you and your sister Judith, right? Was her name? Yeah. And so was it more just inspired by the like a humanitarian or humane orientation? Then
I don't know if it's humane. That's not the right word. I certainly know better than the average Joe or whatever you want in those regards. No, I don't think so. I don't think it's humanitarian. I think it was dictated and just had no choice. I met doctors in Harvard's Dana-Farber Center. My father became close to Sidney Farber, then they named it after Sidney Farber Children's Hospital. And then it became the Dana, after a rich person who I guess donated, Dana-Farber Center. And when I went to see my sister, I saw doctors doing research. I didn't understand it. What's that? What are they doing and why? But then it became serious, and I broke my back playing basketball. And I said, okay, that's that. I was totally depressed. And then I began thinking I better become something other than a nonsense, less than mediocre basketball player, because I'm not going to go any further, right? I mean, I could play high school, but where am I going with this crap? And, I got to get serious. There's more to life than just dating girls and playing basketball. And, my thoughts turned toward my sister, yes. And so it's not humanitarian. It was just like, okay, I had, everybody's different. They think scientists are the same. They're as different as you guys differ. Then barbers differ. Then lawyers differ. They're all different. And personality-wise, I like gambling. I like the dare of it all. I like the upstream more than the downstream. When it becomes practical, you got to do it. The blood test for HIV, we did. You have to do it. But, related to this damn thing and everything is practical, right? But I much prefer if you're presented with a problem it's not going to be newsy. It's not really newsy stuff. It's more like, how do you know this disease is caused by a virus? Why do you think so? And what kind of a virus do you expect? And I always thought to myself that tropism of the virus will tell you something about what it does, meaning the target cell. Mm-hmm. Its strategy to survive will tell you something, and our immune response will tell you something, and then you can see what kind of disease that should fit and start with that. And so it served me pretty well, and I got involved in blood cells, because leukemia. And the irony is, when I was sent to NIH, when I went in a very competitive time in lieu of Vietnam, I wanted to learn technology and research, right? And the MDs had to, because there was no place to go at the time. There weren't centers of excellence in diabetes and this and that and the other like there are today. There were just 3 cancer centers: Roswell Park, MD Anderson, and Sloan Kettering, and there wasn't anything else. I mean, that was it in terms of centers of excellence in something. So you had to go to NIH if you were an MD, chasing the PhDs, which is what I was doing, trying to learn technology. And then 2 years became 3, became 4. But when I first arrived, the guy I'm supposed to work with left to go to Duke. And I got sent. Where do you think? I never wanted to see patients ever again, right? Mm-hmm. I went to the childhood leukemia wards for a year, just finding death. I mean, that was it.
So that's what I wanted to ask, because was it your sister's? Was it that experience, or because of your sister? Because you ended up discovering HTLV, which, if I understand it correctly, is the first.
Oh, I ended up discovering the first cytokine too, interleukin 2, in 1976. Well, my colleagues and I, always there were colleagues, and I did nothing myself alone. I mean, if we didn't have good postdocs and people working with me, I would be nothing. I mean, I was in the right place and I had great people working with me. So that was true of the bulk of my career, certainly at NIH, and right now in Baltimore. The Institute is filled with great people. However, they're not really my lab anymore, right? You know what I mean? It's like the Institute I direct, but I collaborate, really. So that period was an enormously productive period of time. But sure, there was the experience on those wards. Look, I don't want to mention this, but my associate checked into a local hotel in Bethesda with a bottle of morphine and killed himself. Because you're just talking, you think you're hot shit coming out of the University of Chicago and all the Harvard guys and stuff like that, and you're in NIH, you out-competed people, and you're there to solve problems of the world, and NIH in all its wisdom puts me on a childhood leukemia ward where all I had was defeat. It wasn't great. So I never wanted to see a patient ever again, and really never did, except AIDS kind of forced it on me. You can't get away from it. I mean, they're in your face. Everybody, I became friends with a lot of people that were HIV positive. You can't avoid it. But before that, I really, really shied away from, seeing any patients, right? I have a certain passion when I get a little competitive. And then I have the passion because this is too big and too bad, so we need to solve it. But I much prefer to be working, I don't want to say I'm not a quiet type, but I really do enjoy the mystery of disease at its onset. You tell me, look, I think you're going to work for the rest of your life on multiple sclerosis. We don't understand it well. That could make me happy.
What's been interesting, first things I wanted to ask you about is that you're clearly with COVID thinking a little bit differently than a lot of other people, right? I mean, I've heard you talk in some of your interviews about how the bulk of the various efforts to try to develop a vaccine are focused on what you call the spike, right? And the sort of traditional antibody response type vaccine, and you're going in.
Start with it. Yeah, you're right. You're right, Matt. Both of you can start with the. I'm coming across here like I'm trying to throw darts at myself, and I'm not into that usually as boomerangs. But it is interesting to think about it, that I start always with some bias, and the bias being, we don't like to be. However this comes out, it's not going to sound good, but I don't like to be with the mass, with the herd.
Right.
Okay?
Right.
And too often we have herd thinking, and sometimes it's right, and sometimes, and very often, it's not. And when a mass of money goes to certain places for decades at the expense of young people who are creative getting a chance, when you need ideas. See, I feel this way. If it's public health, it's mandatory we are maximum cooperative. If you have an answer in your hand and you're pretty confident that's the answer, you damn well better share it. Mm-hmm. You better be cooperative. When we had the blood test, we ain't going to say, well, we're going to hold on to it, right? Or we're going to, or virus material that we could grow forever for the first time. We had several that we could grow forever. We're not going to say no one else can have it. We're going to just dominate. No, that would be, you couldn't live with yourself. A normal person, I think, could not live with that. So when it's a public health problem, you have to cooperate. You have to collaborate. But when it's in the early stages, don't listen to people who are against competition. It's everything. It's almost everything. I think Jim Watson once said it's everything. It's not everything, but he's almost right. It's a lot. Because at that point, you want independent thinking. Young people today, let's go back to HIV pre-pandemic, but they don't get funded. It's really, really hard. And is there room for creativity? Well, there's not room for much else but creativity in HIV, is there? The problem is solved of cause, of blood tests, of good therapy. Those are solved. What is needed is a vaccine, right, for the world.
Well, with COVID, what was the atmosphere in terms of that competition you're talking about in the very early stages? What were the different directions that people were going in? Because you went into a very unusual direction.
But remember, I formed something. I co-founded the Global Virus Network. Mm-hmm. That was 2011. And Christian Brichot, former president of Pasteur, is the president of this. As you know, you interviewed him.
Yeah.
But we meet on telephone. We're talking to the globe. I mean, we have people from China on the phone. We're talking. Our government doesn't have that. I don't care who in our government, what Washington Post favorite son or daughter or whatever, certainly not the president, of course. But anyway, they don't have that opportunity, and we do. And yet, no one asks an opinion. That's the facts. But we learned very early on. So why I went this direction? But let's forget the personality problem. They don't need somebody arguing for the spike. It's self-evident. I'm not saying we don't need a conventional vaccine. I mean, that's been a little bit misinterpreted. I had to correct. It was a bright woman reporter, but she did have it in there as if I didn't believe in an adaptive immune response vaccine. Of course, I believe in one if we have one. Look, measles is ideal. Lasts a lifetime, one shot, right? How many measles are there? How many viruses do you think we have, or pathogens that you can give one shot, last a lifetime? Well, that's rare. That's rarer than, I don't know what to say. It's very rare. I mean, it's very, very unusual. How often do you think we have a correlative protection? As Paul Offit likes to say, the biggest correlative protection is protection. And the correlates we have, we think, such as neutralizing antibody or killer T cells or this, that, and the other, we sure let people, and still is, because the massive funding is still there for AIDS towards neutralizing antibody. Now, am I against neutralizing antibody? The first paper in HIV against neutralizing antibody is in Nature by myself and Marjorie Guroff, my associate, when I was at NCI. It's like 1984 or something. So, of course we were hooked to neutralizing antibodies, but you look what's in your face, not what you want to believe. So there's massive money for that on both coasts of the United States, massive. At the expense of what we need is creativity right now. And I can only tell you this. I've never seen a monkey experiment where we had protection that it correlated with neutralizing antibody. Thus, we got off the train. said, this is wrong. You know, you got to think of something else. Well, it'll work if you treat with neutralizing antibody. Yes, it can inhibit virus therapeutically. And if you give it just before you give the virus challenge, you can block infection. But try doing it with an active vaccine. That's called passive vaccination. You don't get it. So we don't know how to protect against HIV to this day. All right. So now we come to a simple virus. So how did I start out and what's going on? No, I mean, I'm like you, just reading in the paper and fearful that we're going to all be in trouble with this damn thing. So yes, we need a vaccine, and yes, we need therapy. And no, I never was impressed by Clinton's hydroxychloroquine. Clinton, what did I say? Clinton?
Trump's.
Freudian slip about the nature.
I mean, I have to say my prayers right now. All right, no, I don't know why. I was thinking of something related to Clinton before. So, yes. Well, I don't want to make fun of it because in our institute, Davide Zolla tells me in northern Italy, some of the, they got on top of things pretty well with a horrendous epidemic. People don't realize there were 220,000 Chinese there around Milan because of the fashion industry. And they went back, most of them, for New Year and then came back. So they had a wallop. But they handled it rather well. That's why the reward is coming to them. And part of it was not just social. They claims, right away they used dexamethasone in the sick people. That's all. They use heparin right off the bat when they start getting complications. As soon as they see complications, on goes the heparin. They're using hydroxychloroquine too. So I don't want to stand here as like a know-it-all that I say that can't work or won't work. It wasn't the thing that I was thinking would be so very good. And we don't have data in America to say one way or another that I can be positive of. But let's say it's certainly not a tremendous drug. But nor is Tony's point and makes a press announcement on remdesivir. That's not a great drug, remdesivir. I mean, anybody I talk to clinically doesn't think it's a great drug. I don't think he should have made a pronouncement about it. Where is it now? Why are they talking about it now? Are you writing about it, remdesivir?
A little bit, yeah.
Yeah, a little bit. Okay, and that's where it belongs. A little bit. It's not the right answer, right? I mean, there has to be better things. It's
a little piece of the puzzle, basically, right? For treatment. Yeah.
One of them could be and should be interferon, the innate immune system. Now, wanting to tell you what happened with me, we're not going to compete with those big companies that they decided to give all the money to. I don't know anything of the background of those decisions, but it doesn't take a genius to pick out the spike, right? The virus, the spikes, right?
Meaning the shape of it. That's how it got its name, coronavirus.
That's the guy that comes at the cell, the first thing the cell sees, correct?
Mm-hmm.
So, of course, you target it. That's what we do with HIV. You target the envelope all the time, right? We look for neutralizing antibodies. Wrong. Doesn't correlate. So we have some things to solve there that are still quasi-mysterious, but I think are solvable. And it's different than people are thinking. And if you look at the results of Lou Picker from Oregon, you see the beginning of answers. And Lou Picker's got tremendous data, but we think we can figure out some reasons why that he may not yet have arrived at and move something importantly. But that's another story once again. So let's go back to this thing. So, I'm there on obviously since I founded the GVN, I have to be on phone calls with the GVN, and I'm getting more and more interested in the pandemic. And you say, you can't avoid it. So the whole institute, without me saying, you got to do it, our public health side, who are all over Africa in 7-8 countries, they're still involved in this as well. Our clinic is certainly involved with clinical trials. Kodalil, who's my partner in crime with this, with Chumakov, with OPV. In fact, we just got off a nice discussion where it looks like we're going to get support for a substantial trial from a group. So, and there's 2 groups coming in to support.
Sorry, what's OPV? I don't.
Oh, oral polio vaccine.
Right. Okay. Right.
Right. We'll come to it. Right. Yeah, right. So, we're sitting around talking about things and realize everybody and their mother, there's 160 vaccines, they say. And I said, I bet 150 at least are going to be on the spike. And indeed they are. Now imagine you are a billionaire, Matt. I know you can imagine that. You probably do a lot of imagining at your age. And then you're imagining you're a billionaire now and you're sitting in your office like you are. What does it take if the Chinese on January 10 published the sequence? Could anything be easier? being talked about by NIH colleagues as this is historic. What historic? I mean, this is ridiculous. Historic. It's obvious, right? And you don't know it's going to work, right? And if it works, you don't know how long it's going to work. This is a point I'll come back to. There's no big deal about you ordering that sequence to be made as RNA by one of about 200 different companies on Earth. You can buy it if you got money.
Right.
And when you buy the RNA, you can stick it in the muscle and it'll translate to a protein. And you'll have the spike protein, which will induce antibodies. And you suspect they'll be neutralizing because they might target the envelope. Much, much easier and much better than HIV. So the problem is solved. Day one.
That's what they think. That they
right.
think that's going to be the solution.
But if you look at all those vaccines, they're all against the spike that I know of, with a few exceptions. My wife showed me an article in the Post, and I say, somebody, just another spike damn thing again. I don't need to hear this about this guy in Pittsburgh. I would recommend maybe a little more follow this guy in Pittsburgh. He's relatively young, but he's taken a measles virus, and he's got a way of attaching, spike proteins and other proteins to it. Now, the spike is not perfect for getting T cells, and I'll come back to that in a moment. If you're going to try to get T cells, you don't just generally use the spike, the envelope protein or the outer proteins. But in any case, I said, I'm not going to go back to that guy again. So I figured that out of 160 vaccines, 150 will be just a spike, presented in one way or another. Sanofi, they're purifying the binding site of the envelope, mass producing it, okay? Just the protein. They're not doing the RNA translation. They're going to do standard technology, recombinant DNA, production of the protein, then isolate the binding site and use that. Somebody else is putting it in an adenovirus vector, replicating, another non-replicating, another will be a DNA form. The owner of the Los Angeles Times, Patrick Soon-Shiong, and his biotech companies, he calls me periodically, usually to bitch about something that he thinks he's being unfairly treated about, and we talk. I mean, I know it's a strange world that I find myself in, but yeah, he's a billionaire. I'm not. But we talk. We talk science. He knows quite a bit of science. And he's got an interesting approach. It's a spike plus a little more. We can see what happens. So I said, well, what the hell can I do in that? Nothing. Right? I mean, they've already paid them to do this. And however, we started seeing holes in it. And what were those holes? Well, the first thing I learned was that bats control coronaviruses simply with innate immunity. The next thing is that I read a report from a Dutch group that the seasonal coronaviruses, they couldn't do SARS-2. Let's just call it SARS-2. That's what I always call it. The seasonal coronaviruses, the antibodies to the spike are short-lived. Okay? That's fact number 2. Number 3, we looked at the structure. The structure. Anybody could have done this on January 10th. You look at the structure of the protein. It's a lot like HIV in its glycan shield, sugars covering important sites of the protein. In mannose, a particular kind of sugar, side chains. Prediction. HIV antibodies to the envelope do not last. Why? Because of that. Do we have data for it? Yep. After talking about it for about maybe a little over 10 years, and nobody wants to hear it because you have your candidate vaccine. You don't want to be somebody saying even if you got the best, it's not going to last long. So it's a problem. You don't want to hear that. But eventually, I convinced Tony to listen carefully. And so I had 2 hours with his staff, and we got funded. And in the midst of the funding for that, I can tell you we know why the antibodies don't last when you're glycosylated in that fashion. So we saw this guy is glycosylated the same way. If you look at another protein, nothing to do with a virus, just a protein that has these sugar groups in a certain way, they won't last. What happens is, I will tell you, you don't need this detail, but I'll tell you. You
mean the antibodies in a potential vaccine won't last?
Right. The B cells don't mature properly into mature plasma cells in the bone marrow.
Okay.
You don't get there.
So even if you could develop one that would work, it wouldn't protect you for very long.
I'm going to come. You want practicality, and you got it. That's it. Okay. And then I'm going to tell you something that I heard yesterday. It's not actually very nice. If you don't last, if you're not durable, you're going to get what the Army got. Do you remember the Thai Army trial for AIDS? RV144 is the only trial that worked. Mm-hmm. Nelson Michael, Jerome Kim, who's now head of the Vaccine Institute in South Korea, who had playing a major role in South Korea, and their colleagues. We collaborated with them. They have the only positive data, caused press conferences and so on. However, it crashed. So what was 60%, 65% quickly became 30%. And that was the end of it all. And when you followed their curve, I went to the press conference. I was invited, and I was the only scientist, I think, there other than themselves. And I walked out after about 10 minutes because slide one, they showed the curve. And I said, that's it. I've seen this before. I mean, it's like, here's your protection, and it's right up here. And then as the time is going by, it's going like that. Time after vaccination, it falls apart. Okay, is that what I think is going to happen here? Yes. Do I think they'll get protection? Maybe, probably will. Do I think they'll make noise? Yeah, probably will. Look, durability of an antibody is actually known by 2 to 3 years. That's when you have what's called the gamma phase of antibody development, the formation of the plasma cells. 6 months doesn't tell you anything. But the study from Holland showed that the coronaviruses in general, not including this guy, they didn't do this guy, is 4 months. That's not too good, right? So that's bad news. Then the question will become, do we know there's no harmful effects if you're infected from the antibodies? We don't know that yet. I'm not saying there will be, but nobody knows that. That's something that takes time to observe. You can't talk about a vaccine until you have a vaccine, and you don't know you have a vaccine until time goes by.
And I've heard you talk about this before, right? That when the press and politicians talk about the timetable when we can have a vaccine, it's a red herring when they're doing that, right? Like you really never know until you actually have one that works.
That's what I like to say. You have a vaccine when you have a vaccine.
Right.
In the beginning, in those announcements, I don't want to say are meaningless. They will have some meaning, but then how far you take that meaning is the question. And you have to be aware of that. After 2 years, you'll know. Once they come out with something, then about a year and a half after that, you'll be able to have a nice story that sums it up well for the world. Also, the late Morris Hilleman used to say, feasibility is everything. He made most of your vaccines. Other than rabies and polio, probably all the vaccines you're taking came from Hilleman. And he was on our board, and for years he always used to say to me, the biggest correlation of protection is protection. It often doesn't correlate with anything we measure, and that's a good thing to keep in mind. And the other, he said, when you start with a vaccine, start with feasibility. That means cost, safety, ease of delivery, ability to get it to developing nations.
Okay, so this gets us to the oral.
Right.
So
I'm on the phone. Mm-hmm. I'm on the phone. I go back to being where I was. Mm-hmm. You're keeping me on straight line now. And on the phone, I'm realizing that these things are probably not going to last. Very likely. Now we're seeing evidence for it, both clinical and with the seasonal coronaviruses. Now there's evidence even with these guys. They don't last. All right. So I am also, we're talking to people in China. We're getting information earlier because they're a little ahead of the curve, of course. And then Konstantin Chumakov is at the FDA. He's the associate director for vaccine research at the FDA. He's my partner in this, as is my colleague who directs the clinical division by our institute, who replaced Bob Redfield, who became, as you know, CDC director. And Chumakov's mother didn't get credit at that time. The ladies didn't get too much credit. Made a startling observation. Her husband was a very famous virologist, Chumakov's father. Actually, he's one of the people whose pictures is on my wall as you come in our institute. Sabin gave him credit for the polio vaccine in Russia. Said without him, nothing would have happened. But the mother observed, in the middle of a bad epidemic in Russia of flu, that better than any flu vaccine, people were protected by oral polio. What in hell is going on here? So she published this in 7 papers in the Russian literature, and finally there's one in America that gave a review, 1 paper only, and she observed that 3.8-fold reduction in influenza if you had oral polio that year. And then other people observed it in Singapore, and then other people observed it with measles. The childhood illnesses that were infectious were disappearing. And she called it virus interference. A few years later, the cytokine interferon was discovered. At about the same time, I was discovered what people say is the first cytokine, which was interleukin-2, or T-cell growth factor, getting me closer to this stuff. But I never really got into the depths of innate immunity until now. But I'm telling you how this happens step by step. So we're learning that the bats controlled coronaviruses by innate immunity. We're learning these other things. And then we find that the people who have higher levels of interferon do clinically better. And therapeutic trials with interferon have done rather quite well. And it's just not been talked about. That's the innate immune system.
That's what I wanted to ask. For people who don't understand the difference, what's the difference between...
Innate immunity is what invertebrates have, and we got it.
Frogs, right? Yeah.
Right. Everybody else has innate and adaptive. But the innate immune system, the people with that, and people, the animals with that, like sharks, for example, do fine with only innate immunity. Well, what the hell is it? It doesn't take weeks or months to come, like the adaptive immune system. After your vaccine, when you get vaccinated with the spike, it's going to take you weeks to get antibodies, maybe months to get the right antibodies, maybe a month or 2, and then they're going to peter out, I tell you, in 4 months. And the T-cell response will last longer, and hopefully that'll be the saving grace of these vaccines, that they gave some T-cell response which was not really expected. I don't know if they'll be helpful, but they might be, and that may give the durability, if we have durability. All right, let's go back to innate immunity. Hot area for basic research today. Poorly understood. Interferon is a hallmark of it, the hallmark of it, all the interferon genes. But so this is the enemy is at the gate. The barbarians are crashing through the gate. This is your emergency response.
It's like the primitive alarm system, right?
Absolutely. Okay. You can look at it militarily. The adaptive immune system is the cavalry coming in later, right? Okay. The guy says, the innate immune system says, good Lord, I'm overwhelmed, I can't handle it. Then comes the adaptive immune response. Most infections you ward off just with your innate immunity. You get a little chill, maybe a low-level fever. That's your interferon and a couple of other cytokines. And you're there. You're not triggering off a pound of antibodies or killer T cells, which would take some months. You're better in a few days. How can you get better in a few days from your adaptive immune system? Not doable. So this is your innate immune system taking care of things. But is it more than interferon? Yeah, it's much more, but it's complex. We don't fully understand it yet. And it involves more another lineage of cells, not the lymphatics, but the myeloid cells, cells we call macrophage, monocytes, neutrophils. And exactly what they're doing is complicated and being worked out. Now comes the problem is, how long does the innate immune system last? Answer, we don't know. But it's probably maybe 2 months, maybe 1 month. But you could boost it again and again, and we could extend it out. And then there are people who are finding that there's something called molecular imprinting, which means that the genes aren't changed, but by modifying histones or things like that, that you can have this thing last for over a year. That's what people claim for BCG. BCG is another example.
What's BCG? I'm sorry.
Oh, Bacillus calmette-guerin. It's a tuberculosis modified bacillus that's used for the same purpose.
Right.
I don't think it's good as OPV. We don't think it's anywhere near as good. It's not as it's much, much more expensive. You have to inject it and you get some scarring off with it. And it's not easily available. And we're running out of gas, not having enough of it in the world. Whereas oral polio is easily available, easy to produce, safe as can be. There's not one case in billions and billions of people of a complication if the person has got pre-existing vaccination, as most of the world has today. Before there was a vaccination, 1 in a couple of million people got a polio. 1 in a couple of million. No other complications. But if you're vaccinated, chances 0. Not many drugs and not many vaccines you can do much better than. Cost, 15 cents a dose. Availability par excellence. Can be produced by Biopharma of Indonesia and India. Can be produced by GSK and by Chinese, and by where we got free doses ourselves, Sanofi Pasteur. So, you know, when we're seeing this, we argued last February, March, April, May, June, July. You want to see somebody that's a little angry? Me. Konstantin Chumakov, Chaim Kotolo. You know, we each handle it a little different. They really kind of, you know, yeah. I mean, I'm shocked because everybody was excited when we talked to them about it, and we didn't get funded.
Wow.
So now I can just tell you where we're at. We're in several foreign countries right now that are doing it. That's one. So we succeeded in some foreign countries. So we'll get data. Two, we have a few people interested in supporting us that are more private. What happened in the United States? Well, we didn't start off with nothing. Chumakov's in the FDA. He went through the FDA, got positive reception, but he needed funding before we could submit an IND. My colleague Kotolo didn't want to waste his time going.
What's IND?
I don't know. It's something you need from the FDA. All clinicians know, but not. But even though I'm an MD, forget it. I don't know. It's something that you need from the FDA to when you get like a drug approval. You could say, well, polio is obviously approved. Yeah, but when you're using it for new purpose, right? You got to get approval from everybody. Right. So that's FDA. But everything should be good there. But my clinical colleague didn't want to go forward applying for something until we knew we had funding, right? Okay, so next step is we now say, okay, let's go for funding in the United States and do a big trial. Meanwhile, we have something in England going on for deliberate infection. I mean, some people, including Bob Redfield, wanted us to do it with SARS too, but then he realized now that 30-year-olds were also getting sick, so he stopped advocating for that, and I was never going to do it anyway, because it would be just my luck in the twilight zone of life that I kill a few people myself, right? So we don't want to do that. I mentioned to him his hero, Walter Reed, would be in jail today. You know, if you know the story of Walter Reed, I don't.
What did he do?
Oh, yellow fever. He proved it. He saved things for America on the Panama Canal, but he killed a number of soldiers in the way, volunteering to get bitten by the mosquitoes that he knew were transmitting. So, you know, you don't do those things anymore. You don't chance those things. But we have a place in London that will do flu, again, just approved in modern times, and will do the seasonal coronaviruses, which should impress people because there's a lot of homology between those viruses and SARS-2, but they only cause common cold. So we're ready for that, and we're looking for fundings. We know many people in the U.S. Army are very interested, very excited, in fact, but hard to them to get to the right, you know, they tell me any GS-13 at any level in the government can block it if it's not fitting exactly on the line of what they're funded for.
So, sorry, is part of the problem here that OPV is just too cheap? Like, in other words...
Well, I'm going to come. You're getting one step, almost done. You're getting one step ahead of me.
Okay.
The thing of deliberate infection, where we could prove things in a matter of a few weeks, would be cheap. It would, instead of being like $40 million, would be maybe $2 million, maybe less. We could get answers in 3 weeks' time. We still hope that happens very strongly. We got a lot of people interested. And let me tell you, backing us and consulting to us are, can't do much higher, of polio experts that we have on our committee. So we have the polio experts, both at WHO and in the United States, head of committees. We have Stan Plotkin, who's an enthusiast, who's about the most experienced vaccinologist in the United States. And you shouldn't make any opinions without checking with him certain times when you're writing on vaccines. Stan Plotkin. So the main thing was to do a trial in the United States. So we took a variety of GVN centers, Global Virus Network Centers of Excellence. So there was the three Caballeros, the three of us, plus we would handle Baltimore, Washington area, and then Roswell Park and University of Buffalo, and then Cleveland Clinic. And then lately, University of Nebraska wanted to join in. So we figured that's enough because it's going to get way too expensive. But meanwhile, the rate of infections were coming down. So we need more and more people. The cost is going up. I can tell you CDC, we talked to Bob Redfield, who I obviously know. He was very excited by it. So we said, that's round one. I talked to Tony. He was there at the same time. I would like to say he was enthusiastic. I won't say excited, but enthusiastic. We got site visited by his institute. 14 people came on the screen like this. I can all say they were all, unless they're great actors, really excited by it. And then 2 weeks went by, we heard nothing, and I called Carl Diefenbach, Tony's right arm, and he said in the end it wasn't received favorably at the high level. That's the end of the story.
And why do you think it wasn't received favorably?
I don't know.
I mean, I guess the question I'm asking about.
Let me answer part of your question. I'll tell you what Tony first said. He first tried the safety gangle. So I wrote him a long letter and said, this is, I don't even know how to pronounce the word, R-U-S-E, a ruse. I didn't say by him. I said, it's either ignorance or it's a fake. And he said, let me tell you about that. The water supply, my ass. Okay, I'm sorry, I'm quoting what I said. Mm-hmm. And I said, this is, what are you talking about? Water supply. I don't mean I said it to him, but generally discussions. I said, water supply, America hasn't had a problem with water supply in about 150 years, but let's say something more. The whole world is receiving oral polio, the bulk of the world, billions of people. Now, Indonesians, Chinese, Pakistanis.
Just to clarify, were they saying that there was...
Yeah, they brought that up as one possibility, right? They brought up possibilities of why they were worried. So the counter is, the world is getting it. Do we stop Israel? They get it. So do we say no Americans can go to those countries? Do we say that when they come to our country, they can't use our toilets? Do we say they can't drink our water? What are we talking about? They can't go to the men's room? I mean, the whole world comes here and we go there. What are they talking about? And also, in case you weren't aware, between 1962 and, 60? Yeah, and 2000, every American received oral polio. You received oral polio, I bet. It used to be a sugar pill. Now it's just a pill on your tongue. That's it. Couldn't be easier to take. If I go back to work, I'm going with polio tablets in my pocket. What's the harm? We all get vaccinated against it. There's no case of polio of any kind if you're already vaccinated. I'm vaccinated. And my chances would be about 1 in 3 million anyway. So, I don't want to go back to work in a crowd and go to restaurants until I have oral polio in my pocket. And it's going to work right then. In a matter of 2 hours, I'll be fine, right? I go into a crowded place, but I take a polio tablet beforehand, I'm okay. And it should last me for, I don't know how long, but it'll certainly last a month. And I think it'll be significantly longer, months plural, and then we would boost. So now it's getting a little bit late. It's going to be 7 months since we've been arguing for this. It's, February, March, April, May, June, 6 months. So, that's it.
I mean, I guess the question I have is, so you take a drug like remdesivir, it's going to be $3,400 per course of treatment, and you have oral polio, which you're saying is 15 cents. Is it possible that there just isn't a lot of momentum for this kind of treatment because companies don't see the possible long-term financial benefit?
I need a consultation. I need to ask Dan what he thinks.
Yeah, I'll be the expert on that.
Yeah, I mean, honestly, Matt, I don't mind a politically sensitive issue, and I don't mind getting attacked by somebody because surely it will raise things against us. But I got to watch out of hurting colleagues also, and hurting the project. Mm-hmm. I don't know. I truthfully don't know. It's a bad thing to say.
Right, because we're on the verge of a whole bunch of companies coming out with vaccines, right? Or and
you're right.
I've heard some doctors say that there's a possible future where you'd have to take overlapping vaccines, where it wouldn't just be one that would be. I mean, what do you think is the most likely outcome, like 2 years from now? Is everybody going to be taking a vaccine? Is everybody going to be taking OPV? Is there going to be
Can you imagine how long it will take to work out with overlapping vaccines to get answers?
I mean, yeah.
You're going to be confused, and there'll be easy. Let's take the most cynical route here. Mm-hmm. I never heard of such a thing before in my life, right? Let's we mix up a lot of things and see what happens. These are not things like a drug trial where you can get answers by measuring the titer of virus like in HIV-infected people, where combine A and B and you can follow it 2 days later. Get the virus titer, get the CD4 count, right? Mm-hmm. If you're going to make comparisons to things like that, that's a joke. You're going to have to wait a long time to find out what works best, right? You're going to be sick, and everybody can be home. You know, they're long gone. You won't even remember their names, the companies. I don't know. I don't like the way that sounds, but I'm not in a position to say that it's not rational. I am in a position to say I'm surprised, and it's going to be hard to interpret, isn't it? Anybody could say it's going to be hard to interpret. If you got a certain amount of data, and then you say, okay, now I want to use this other vaccine, and now what is causing what? And if it works less, is that because of the second vaccine? Let's try a third one. Let's go all around America. Maybe the top 5 can all be rewarded. You want to be a cynic? I mean, if 5 companies got $1 billion each, I don't know what they got. And now Close. They all want to, we can't get 50,000. I mean, really? Well, we didn't get any money, right? We didn't even have a chance for a second comeback. I said, can we come back and just destroy the arguments that you made about safety? And then when I made the arguments, there was no comeback. Yeah. You know, we ended up.
Does the United States have a great system for developing necessary life-saving vaccines and medicines? Like, if in the ideal world, how would we improve the system that we have?
I think we really need to bring back a real good public health system that was dismantled. And I think there's Terry Lehrman is my board chair for the Institute of Human Virology. He's a Democratic operative. He's Steny Hoyer's chief of staff. He operates at that level, and he's involved with Biden now and so on.
Can you tell Biden about Medicare for All?
Sure. I have no contacts. I mean, I can tell Terry Lehrman can say what things. He always does things when he needs. If I need a little political help, sometimes he's there. You don't like to do that. So far, we haven't done that at all. But anyway, Terry and I are it's already started combining forces on some kind of article like this for an editorial somewhere. And maybe it should be written by pros. We're not pros in writing, and maybe just turn it over. But we think, you guys could do a great job with something like that. Yeah, he wants me to maybe write the science part of how things could have gone better. Well, what the hell did I form the Global Virus Network for? And why hasn't it been picked up and helped by anybody at NIH or elsewhere? We're now doing okay because, companies are starting to donate and things like that. But we went since the 9 years we formed it, until a few years ago, we were operating on shoestrings and everybody donating time free to it, like myself, like Matt Evans, who's a Madison Avenue guy in New York, who's secretary treasurer, and he works at least half his time for nothing for GVN. Christian Brachot was getting peanuts to be president. Now he's paid, partly by University of South Florida, but now we have enough to have a staff of about 10 people. So, we're now moving stronger. But here's a collection. I mean, maybe someday, you talk to Christian, but, where's a collection of, we put down who we think are the greatest virologists on earth. I'm tired of reading the greatest infectious disease doctor every day in the paper, The greatest, there isn't a greatest infectious disease doctor. There are very good infectious disease doctors and maybe some great ones, but they're specialists. So we took the virologists to try to cover who's the best in every single kind of virus on earth. Who are the best ones we could get? And then we add on to it and add on to it and add on to it. And there's criteria to get membership. There's criteria to be a center of excellence versus an affiliate. Now I can we can go bring you into an office and see a map, and you can say to me, last week, a new epidemic of a gastrointestinal diarrhea that's killing some children developed in Egypt. And help, you can say. We have WHO. WHO is surveillance. Good. We don't want to do surveillance. WHO, if they know the nature of it exactly, they have a group that they can telephone. When it was AIDS, they didn't know what a retrovirus was. Most people didn't. They were paralyzed. WHO was paralyzed. So was CDC, actually, off record. Well, no, not off record. CDC was fairly paralyzed at the time because they didn't know anything about retroviruses either. So you say, now we have experts on everything. I press a button, I'll get a light. Really, we can't afford the lights, but we'll get, we'll know. Where in the world we have the best gastrointestinal experts on earth? Let's say we take 6 of them. And we say, you 6, concentrate on this. Do a report. Tell us what you think. Now, if it takes money to send them there and to work, we need help usually, right, at that level. But we have held annual meetings, an annual course for underdeveloped nation people in virology in Baltimore, and an annual meeting somewhere on Earth, moving. It's been in Russia. It's been in China, for example. It's been in a lot of different countries since 2011. We have every 10 days an international call with everybody on the phone from Australia, Singapore, China, Europe, and East and Mid United States. Then the West Coast and I forgot what other part of the world gets another call from Christian that I'm not involved in. So I get up at 5 a.m. to be on the 6 o'clock call every 10 days, and we review everything on Earth. And why hasn't the world grabbed onto it? But it stuns me that the world, everybody's saying, we don't know who to ask. I say, oh my God, if that's the biggest crisis we have, how can you listen to your government when every government differs one from another? Doesn't that say it all?
Well, that's another question I wanted to ask you. Compared to the response to HIV, has there really been a lot of progress towards creating that exactly that kind of global response to?
No, it's the opposite. Problem? It's the opposite. I had an editorial that I was going to send to LA Times that Patrick asked me about, and it was, what does the word pandemic mean? It means all, right? Mm-hmm. A pandemic has to be all in together, girls, right? I mean, if you're going to jump rope, everybody's in together. Remember that thing? Everybody has to be together in it. For God's sakes, this is something that you can't have one country doing one thing, one country another. There has to be a coordination. U.S. has to get along with China. There is no choice. I mean, I'd like to be a tough guy too with China in some respects, right? Not by harming Chinese American authentic postdocs and students just because once they had to serve military service in China, they're suddenly spies now, which is absurd. It's another story for Rolling Stone, right? You got a lot of good stories coming up. Let me be editor for a day. I changed my job. You take mine and I'll take yours for a day. So that was the argument for it. Pandemic means all. That's what the title should be. And we don't have that with COVID. All of us together. We're not having a unified global response. Oh my God, no. And if you say, well, I have to follow the government. Well, go to Sweden, you follow Sweden. And then go just a little bit south and go to Germany or North Italy, be the opposite, right? I mean, so, But was there that with HIV/AIDS at the beginning? How long did it take? No, I don't think so. Well, no, I think, it's just a matter of helping people who needed help. The variation was how much money you're willing to give. And until George W. Bush, there wasn't much, right? I mean, he's the kind of the hero of the money aspect. Our institute is a major recipient of funds for helping Africa since it started, PEPFAR started. So we are in 8 countries in Africa. We were in Haiti. We were in the Suriname, whatever it is. Suriname, yeah. Suriname, yeah.
I don't know how much you want to avoid the politics question, but how does Ronald Reagan's role in HIV/AIDS compare to Trump's role in Corona, in COVID?
What I can say about Reagan is, honestly, I was too busy to notice, and I never was hindered. I want you to know that. I was at NIH. I was at the National Cancer Institute. I was always left alone. I can't say I get all the money I wanted, but Vince DeVita was the director, and then came Sam Broder, and both were very supportive to me. In fact, I don't like to go to meetings and things like that, and I was required for this, that, or the other. And DeVita used to intervene and say, leave him alone. He's going to find some things and things like that. He respected that. And DeVita was a chemotherapist in cancer. And he could have taken the pathway that I had no right to be working in this problem, really. I mean, I'm using taxpayer money, right? But we could argue there was Kaposi sarcoma and there was lymphoma. So there was cancer with HIV. Right. And he knew that I kept insisting it's going to be a retrovirus disease. And the NCI was the only institute, not Tony's, that had retrovirology. They didn't have anything in allergy infectious disease. It was all in NCI. And I had the most significant group, actually. It fell into our lap, honestly. We had T-cell culture conditions. We had discovered interleukin-2. That's T-cell growth factor. So we had the ability to grow T-cells. What's AIDS? The clinicians tell us, they made the contribution, it's a disease of CD4 T-cells. What does interleukin predominantly do? It grows CD4 T-cells. Wow. Number 2, I'm doing retrovirology that causes leukemia, but if they vary, they can cause immune disorders and repression of bone marrow. And then Jim Curran comes and gives a lecture. He's now dean at Emory School of Public Health. He was CDC. He could not do any virology, but he was clearly thinking infectious disease when a lot of theories were not. You guys don't know that, but many of the theories were immunological complex theories and all kinds of stuff. But, a specific bug, there were some people thinking that way, but not that many. And I'm thinking a specific bug and a type of bug, a retrovirus. And Curran gives a lecture, and we're not working on it at all. I'm working in cancer. And at first, there was like 6 guys in New York, 6 gay men in New York with AIDS. And so I'm not going to change the field of cancer research to that, which has haunted me sometimes because unfairly people quoted it as I wasn't interested because it was gay men. My God, it was because there were like 6 gay men, and I'm not going to change from leukemia research into working on something that involves 6 people. But when Curran came back a second time, it was clear this was major. And then he said, where are the virologists? And with all modesty, I thought he was talking to me because he was looking at me. And I said, okay, walking back to my lab, that he's right. It is a virus disease. And what did he just tell me? He said that the risk groups were Haitians, the loads of human retroviruses in Haiti, we knew that. Heroin addicts, drugs, blood. Blood transfusions, blood again. Sexual promiscuity. Well, the HDLVs are transmitted by blood, sex, and mama to baby, which he also pointed out that babies of mothers were having it too. So, a lot of people said, well, I get credit for, like a creative thought. I don't think it's very creative. I think it's self-evident. If you're me, you got T-cells in your hands in one hand, you're working with retroviruses, and this guy gives you the, CD4 T-cells, and plus now the risk groups. That's perfect. So we started.
With COVID, we're similarly at an early stage of trying to understand what this disease is. Are there similar obviously this is nothing like HIV, which was very hard for people to understand. It was a retrovirus, which was new in itself, right? This is a much more of a known quantity. But are there particular qualities to this disease that you find unusual or mysterious in any way?
Yeah. There's not mysterious, but certainly unusual. But no, this is a simple virus compared to HIV. Much simpler, much infinitely easier to find. Obviously, it's not really kind of a discovery. It's in your face, right? You scrape it off your tongue and it's in your mouth. An acute virus infection is usually pretty straightforward, pretty simple, and the genomes are usually pretty simple. This one's a little more complicated. It does a lot of, out of one big stretch of genetic information, it chops up a lot of different proteins, like any RNA virus, it mutates. But you would say basically it's a cytopathic virus that's common. They kill cells that they invade. So it's not mysterious. But what is strange is the extensive diversity of the disease organs that it hits. And a great deal of that has to come from undoubtedly its receptor. Because the receptor is in the endothelial cells, you've got a lot of tissue targets for this virus. The shock was the blood clotting, for me anyway. What the hell? Blood clotting. That's a major part of pathology now, is the blood clotting. As I mentioned to you, the Italians use heparin in anybody who's getting sick, right off the bat.
Right.
And they seem to be doing better than we're doing in our patients. So, I try to follow that as I can on within the GVN. We don't do that. We wait until after the fact, it seems. But they're doing very early treatment with heparin. So that was definitely a surprise. I didn't expect the central nervous system or the cardiovascular extensive abnormalities that are being picked up now. That's a surprise. After that, what's surprising about an acute virus infection? Its only other thing I can tell you is its chief characteristic. Its magnitude of contagiousness is really quite spectacular.
That's rivaled only by something like influenza, right? Basically, right? I mean,
that's right. Influenza. I mean, maybe the bubonic plague. I don't know. Smallpox, right? Smallpox.
So to bring it all back, we're at a stage with this where we're about to hear all these announcements. We're hearing about drugs like AZD1222. There's all these sort of hopeful announcements that are coming out. Would you just caution people to? Is your idea that people should just pump the brakes a little bit on being very, very excited about an announcement that might be coming?
After soon we're in the. Well, we are. China announced in December or January. You know, before long we'll be at 3/4 of a year. With modern technology, I don't think it's anything to brag about that we have some drug candidates coming.
Right.
Why were people announcing about drugs? You want to try to do it with a little bit of humility. It isn't great speed that you're talking about. I don't want to say this, quote me, but even the name Warped Speed Committee.
Right.
It's pompous unless you leave out one word.
Right.
Speed.
Warped Committee. Yeah.
I did say that to them when I talked. And don't wait, the committee, the scientific advisors were laughing. But I said, "What is the Warped Committee?" You can't help it. I mean, be a wise guy sometimes. But isn't it surprising we don't have an antiviral drug after we've gone through HIV? HIV was historical, right? There never was a therapy against a systemic viral disease because it was not thought to be possible. That's because they don't have metabolism of their own. But then the age of molecular biology came, and when HIV came, you could think molecular biologically that you knew stages of replication of the virus. Therefore, you could target with laser speed. Or laser, not speed, but laser pinpointing.
Precision.
Yeah, exactly. With laser precision. You know, an antibiotic, you're hitting a bacteria's metabolism, right? It's a big bomb. But, you know, what do you have to target with a virus? Nothing. Well, you thought. But then when you know its steps are several, you can try to target what's key to the virus more than key to the cell, right? And that's what happened with the breakthrough, which was not the cocktail, which the media got terribly wrong. The breakthrough, I mean, I don't think anybody wanted to tell them that, but the breakthrough was not 1995. And the clinical trials that captured Time Magazine and everything else, although that's what people would start to feel so much better with, but the breakthrough was 1985, was AZT. AZT showed for the first time in the history of medicine that you could treat a viral disease with objectivity, showing virus decline with the systemic improvement. CD4 T cells were rising, so you had objectivity. And that brought in the pharmaceutical industry for the first time en masse.
With the mutations of this drug, have they...
With the virus.
I'm sorry, with this virus, yeah. Have they worsened or bettered the outlook?
No one on Earth can yet say that I know of. Okay. If they are, they're not speaking at the GVN meeting because we hear from everybody on Earth, including, you know, Chinese and the Singapore, with a lot of experience in my own lab itself. Davide Zella, Z-E-L-L-A. Zella is following this like a hawk with his colleagues in the University of Trieste in Italy. And I'm getting lots of reports regularly. And we see hints that some things may be correlating with making things worse. Yet, for example, he has some data that the protein known as NSP1, non-structural protein number 1, which is important for the virus's ability to cause disease and to escape from the innate immune system, is mutating. and that 1% of the strains now have that mutation, which should make it less lethal. So what I see, it's going both directions. Things are getting, some strains are emerging less lethal, some strains are emerging that could be more lethal. And it depends who wins in the end. So far, there's not a clear pattern of how it's happening. But like any RNA virus, this guy mutates. Not as much as HIV, not nearly, but it mutates.
And is it possible that it might end up being a thing like influenza, where they're going to have to come up with a new guess every year?
Yes,
okay.
But I think it won't be because so much of the mutations as it will be the durability is poor.
Okay, right.
So I think they're going to be. I believe. I'm giving you an opinion, but it's an opinion based on what I told you were facts. The seasonal coronavirus, that's fact. It's in the literature. Okay. So that durability angle from the seasonal coronavirus is 4 months, max 5. So that's one. What we see in the literature of people's antibody titer to the spike, compatible exactly with that. So, I would say the structure is more hypothetical, but yet it's what we see. If you see this pattern of glycosylation, and George Lewis, we're sitting here, my colleague, immunochemist, he would be more vigorous about it than me. He would say, wow, shit, this thing is obvious, and this guy can't see in front of him. This is not going to last. It can't last. He would be very vigorous about it. We can't say we know. That's more hypothetical based on what we've seen before. Could this be an exception? It could be, and I could be wrong. But I think when you put these things together as each of them as fact, you got to say that the odds of this not being durable are really significant.
Is there a possibility that using OPV in conjunction with one of these other vaccines that are in development, could that also be a possibility?
That's what we were really offering, a stopgap to get you back to work. And then hopefully they'd have something that then extended. Now, and if that got into a problem, we could come back with this.
I see.
Of course. Why is this avoided like the
plague?
I don't know. You raised the question. I don't want to think that way, but I don't know. I just don't know. But there's other things that are going to come out pretty soon too. Denmark is doing Guinea-Bissau. They're doing Colombia, South America. You won't believe it, but Iran is contacting us throughout this thing because of the GVN, and they're doing 130,000 people in Iran.
A trial?
Yeah. And Indonesia will be doing things, and Russia is already 7 weeks in. But Russia is
This is with OPV?
Yeah. All I talked about was OPV. We don't talk about it because there's no data in our hands yet. Russia's in the wrong province, though. That's in an area where they're hardly getting any infection, so I don't know what we're going to get there. And then there's Mexico and Brazil talking to us, and that's where we are. And then in the United States, I don't know. I just don't know what will happen. Maybe with one of these wealthy foundations or trusts, maybe something will happen. I don't know. Well, it is puzzling.
It sounds like you got some exciting things going on. Kate,
it's not the only thing I do, but I tell you, getting money really takes a lot of time. And this rich guy today on the phone said to me, this is all political crap. He says, you got to get used to it, Bob. This is politics. It's the way it is. This is the way he was looking at it, telling me, get over it. It's all politics. It's usual. And I honestly said to him, it's not usual for me. I'm not used to this. And I haven't had this kind of thing before. I was at NIH, and when I left NIH, in NIH, I always had support. You asked me about Reagan. I didn't answer the question. So I will answer the question now. I didn't have any troubles. I got support. But I had Vince DeVita, NCI director, and he believed in me. So I never had trouble.
But what
do I think about Reagan versus the current? Well, I think Reagan's a nicer guy. That's what I think. He had a much nicer smile. I trust him more. Well, if there was any difference in Reagan, the problem of ideology and the blood test would not have come one day earlier, no matter what Reagan did. Let's start with that. We were supported. We solved the problem of the cause. We made the blood test. Not Allergy Infectious Disease, not Tony's Institute. We did it. NCI, my lab, period. And with the funding that we had, right? And we never were frustrated on funding. Group in Paris had their own funding. We were connected for quite a while. Okay, we had the patent problem. But That's the only place I would say they left me dangling, and I lost 4 years. That's another story, but I lost 4 years of productivity. The key period of my career was lost. 4 years. I would use vulgar words right now if you weren't there. Yeah, you know what I would say. 4 something years.
Yeah.
Were lost. And it's hard to live with that for a while. The anger level was off the charts, and et cetera, et cetera. However, and the government just let you hang there. That was true. But Reagan was by then gone. So I don't even hold Reagan. But you still have an interesting question. How about for the rest of the world? How about for the social issues? How about for the educational issues?
Right.
How about for the hospital issues? How about for taking manhood, womanhood, whatever you want to call it, and saying to surgeons, you can't stop patients with AIDS coming into the hospital, which I saw. They blocked them. I said, this is not contagious across the, you're not going to get it from somebody, sneezing at you. This kind of ignorance was going on too long.
Right.
Right. So that could have taken presidential leadership. What I'm saying is our lab at that time was not hurt. It was hurt later for different reasons. And, it's for Trump, I don't see how you can compare him to anybody. But why do we use governments, period, is my question. Forget Trump. Would I listen to the president of Sweden? Do I listen to any president? Why would I listen to a president of what, health? And why do I care about his closest advisors? That's our national advisor. And so what? I will do just as well going to my own university or Baltimore. Just let me take the city of Baltimore. I think we have public health there. I think we have Hopkins at the University of Maryland. I'll get my advice there and on the phone with the GVN. Jesus. Why do we think central government is going to be expert? Since when do we have major laboratories of research in the central government and downtown in Washington? And when Bethesda, where do we think all this is happening? There are individual labs doing individual research. We have a whole nation of experts, all over the place. Maybe we don't have a way of bringing them together.
Well, we hope your work with the innate immune system produces some results. I'm really anxious to hear what the results of all those trials are in those countries. That sounds really promising, and we definitely wish you luck with that. And thank you so much for coming on. Katie, do you have anything in this?
No, just what's the time? What's the timeline? When are we going to be?
Honestly, I don't know. Ask my friend Tony.
Okay.
Okay, we'll do that.
You can even say I said to ask him that he knows the answer.
Thank you so much. And all right,
it was fun. I enjoyed it.
It was great.
Right.
Okay,
take care now.
Doing your good job. Right. Bye.
Wow, that was a lot of interesting information.
Yeah, he's so you might need a glossary for some of that.
Yeah.
But look, his basic idea, I think, is really an interesting one because we're on the verge of hundreds of millions, billions of dollars worth of research coming to quote unquote fruition. There are these companies that are coming out with vaccines, and they're going to be announced with great fanfare, and the vast majority of them used a strategy that he described that might have been a little hard to follow, but it's like sort of the traditional idea about how you fight a virus. You create a specific antibody to fight the virus, and he and a few other people have gone in this other direction that focuses more on like the primitive immune response, immediate one. And it sounds like that could be an interesting idea if anybody could actually get money to fund it. I don't know. Yeah, and obviously for viewers and listeners who don't know, he was involved in a controversy, a pretty significant one, in the 80s over the question of like who actually got the credit for discovering HIV, and we'd be remiss to not mention that. But look, all the same, he is
regardless of whether he was the one, the guy, or one of many, or someone else was the guy, the French, whatever.
That was a huge controversy for the scientific community. Right. I think for the
civilian population. For
the civilian population, the concrete result of all of that stuff was that somebody discovered the virus. a bunch of people were all working on it relatively at the same time. The notion that it was a retrovirus, I mean, he talked a little bit about the mistake they made in thinking that it was another version of HTLV, which was the one that he discovered, how that got them on the wrong path for a while.
It was they were cousins, not brothers, right?
Right, yeah. And then, you know, the fact that they discovered a blood test early, you know, probably saved a ton of lives. And, you know, I mean, the thing that he talked about with AZT, which I never knew before, which I thought was really interesting, was this idea that, you know, even though the drug itself didn't actually cure AIDS, the fact that they got a drug that allowed them to concretely measure the level of virus in the body, and to sort of take this quantitative approach to looking at how sick a person was, that was the big breakthrough. That's really interesting. I'd never heard that before. So that's obviously not going to be the situation with COVID, but it's just interesting the way that people like that think about these things. The question really is, like, are these drugs going to be, are these vaccines going to be effective? And is there a cynical motivation in just producing something that, as he, you know, I mean, his whole point about durability is like kind of terrifying, right? Like, what if some company comes out with a vaccine and somebody declares victory, which there's definitely going to be like a political temptation to do that. And a whole bunch of people take this vaccine, and then 3 months after that, they start getting sick. Like that's something I don't think that anybody's really thought about that as a possibility yet, but that could happen.
Yeah, you got to write about it, Matt.
You know, somebody
like the biological equivalent of Trump winning.
Right, exactly.
And I thought it was also interesting how inherently political this stuff is, like how there is a kind of universalism required in the response. There is. I mean, you can't really, like, nationalism doesn't really work when you're fighting contagious diseases.
Sure, yeah, no, that's true. I think that's always been true of science, though. The scientific community has always viewed itself as being international. The issues are really logistical, right? Like, how do you get funding? Who does the funding? What kind of bureaucracy do you have to go through to do your research? It is interesting that all these years after HIV, which was like a global phenomenon, we still don't have anything like a global, you know, I mean, they're trying to approximate something like that with the Global Virus Network, but we're not tuning in every day to hear a panel of the 10 biggest scientists in the world telling us what the progress against the disease is, where everybody in each of their own countries is turning into their own political leaders, which doesn't make a lot of sense when you think about it.
Right.
'Cause what do they know? It'll be too bad if they don't end up with some kind of system for dealing with these issues that's better than the one we have, that's fragmented and unfortunately led by us, which has this stupidest research system in the world in terms of how we fund things. And that, you know. All right, excellent. We'll see you all next week, and we have an interesting guest next week as well. So talk to you then.